Students can also later on listen there, can take the benefits. And today, the topic of our seminar is about biofilm, a threat to food industry. Our speaker is Dr. Asma Sohail. She is serving as an associate professor in the Institute of Food and Nutritional Sciences. She earned her PhD from University of Queensland and serving the Arid Agriculture University since last 20 years. Today, she will deliver our seminar. It is about 30 to 40 minutes. Then we have 5 questions. Any participants can write the question in the chat room. Even after the presentation, we will open the house. Now, I give the floor to Dr. Asma Sohail for the seminar presentation. Dr. Asma Sohail, please on your mic and your camera because we are doing the recording of this seminar. Thank you.
Bismillahirrahmanirrahim. Thank you very much, Dr. Azam, for my introduction. And I want to express my sincere gratitude to everyone for taking out their time for this talk. I'm going to share my screen. Could you enable me, please?
Madam, sorry to interrupt you. आपकी आवाज थोड़ी सी कम आ रही है. Please, वॉल्यूम को थोड़ा सा adjust करें. आवाज कम आ रही है. और मजर प्लीज शेयरिंग भी डॉक्टर आसमा सोहेल को अलाउ कर दें स्क्रीन शेयरिंग. थैंक यू.
Am I audible now?
Yes, please also, madam, आपकी screen is okay. Please open your cam if easy. Please. Thank you.
Okay. Thank you, Dr. Azam. Today, my talk is about biofilm, which is a threat to food industry. These are the 3 main topics which I will discuss under my talk. Firstly, I will introduce what a biofilm is. Then I will tell you about the impact of biofilm on food industry. And lastly, I will tell you some of the control measures which we will take to control this biofilm. So biofilm is actually a thin layer of microbial slime and protein which adhere to a surface, and then they produce a matrix of extra polymeric substances. We call it as EPS. It is a sticky or glue-like material, and not only one type or maybe the multitude of cell genera can be present in it. This EPS can protect the bacteria, and it is 200 times harder to kill. Bacteria will continue to grow until the biofilm is disturbed and free bacteria are released, and those free bacteria, they are then transferred onto other surfaces, and thereby they are a constant source of contamination. This could be done by hands, gloves, clothes, cleaning clothes, or other materials which are present over there. This is another figure which shows about the different steps which I already mentioned in the clip before, that the bacteria in its planktonic cells may attach to the surface, then forms the microcolony. Later on, they mature, then they disperse, and again comes back to attach to another surface. Now I want you to show this figure. It is very interesting to know that you can see the image of a toothbrush. And if you see the bristle of a toothbrush under the electron microscope, you will see a network of filamentous substances, which actually shows the biofilm. This could be Streptococcus. In the second one, you can see this pinkish material at the base of the tap. Again, zooming in, and you will see under scan electron microscope, it could be another bacteria which forms biofilms, and it may need minerals and other substances from the water. Lastly, a toy, and inside the ducky toy, you can see the black spots or deposits, which is another dense network of elongated cells. Here, it could be fungi. So you can't see biofilms, but bacteria are present inside biofilm everywhere. Once you know that bacteria can form biofilm and the biofilm can protect the bacteria inside, then we should also know that bacteria can communicate with each other. That mechanism of communication is known as quorum sensing. It is very interesting because the signals they are produced by the bacteria, which is you can imagine as the words. Those signals are actually the words of the bacteria, and it is collectively a system of stimulus and response. Shortly, I will say that bacteria releases the signals, releases the molecules, the words, and then they use those molecules to see as a proxy so that there are other cells nearby. And once those cells recognize those signals, they come, interact, and form the part of the biofilm. Now, biofilm gives protection to microorganisms, but it is a challenge for collective resistance for the food industry. Biofilm caused 60% of food-borne illnesses because they couldn't be cleaned by a simple disinfectants, and they are a constant source of pathogens. However, biofilms give resistance to bacteria, like by giving a physical resistance, like against desiccation, or chemical protection, like 10 to 100 times more resistance to antimicrobial agents or biocides or disinfectants. And lastly, bacteria can stick and form biofilm wherever they require moisture, nutrients, and surface like natural materials or stainless steel, plastics, polyethylene, polypropylene, wood, glass, rubber, and all such type of materials they do present in the food industry. In the figure, I want to show you that these are the clusters of Listeria monocytogenes, and they form biofilm on the stainless steel surface. And if I just show you in this short clip that within few hours, bacteria can multiply a lot in logarithmic form. So they multiply a lot, and once they multiply, and even inside the biofilm, it is a threat to the food industry. Now comes resistance to antibiotics. How biofilm actually resists these antibiotics? There are three hypotheses. The first one is the slow penetration. You can see blue and yellow dots. They shows the antibiotics or biocides, and most of them, they remain outside the surface because biofilm layer does not allow it to diffuse inside. A very few molecules, they diffuse inside, and then they penetrate the surface layer of the biofilms. The second part is the resistant phenotypes. In resistant phenotypes, bacteria in a protective phenotypic state does not allow the bacteria to show its effect against the antibiotics, and the cells are resistant to the diffused antibiotics. The third one is the altered microenvironment. You can see a black color dot spots here, as well as orange background, which shows waste matter and nutrient depletions and the actions of antibiotics, which are whatever antibiotics is reached over there, and the action is actually antagonized. In this slide, I want to show you a research paper from one of the researcher who did their research and showing that you can see in the maroon box that only 2 log reduction was found when the 1000 ppm chlorine was given was applied to different type of bacteria which already are protected by EPS. Now, when the bacteria, they are in the planktonic or free state, only 10 ppm is required for 30 seconds period. And in this case, 1000 ppm was applied for 20 minutes, and still bacteria, only 2 log reduction was found. So they are not resistant to antibiotics, or they are not resistant to such type of chemical treatments. Now, I will now move towards my second part of my talk, that is biofilm and food industry. In food industry, the major concern is about the continuous recontamination of foods due to persistent bacteria, particularly when they are present in biofilms, in all type of lines, like in the processing and packaging, through contact with equipment surfaces, wherever they are. So rule of thumb, where there is water, there will be microorganisms. And bacteria, fungi, they all can thrive under the warm and humid continuous conditions which are found in the cooling systems in our food industries. But bacteria is particularly problematic because they attach and form biofilms. Once they form the biofilms on the surface of heat exchangers, then it limits their ability to transfer and reject heat from the water. It also could block the tubes, plug the filters, and it could also damages the surface, like metal corrosion can occur, off flavors, off notes of our products, food products occurs, the whole batch becomes defective, shelf life reductions occurs, and the food industries receives lot of market returns and complaints. So what are the main sources of contamination of the food industry? Biofilms mainly cause problems in dairy, meat, poultry, seafood, vegetable processing industries. And there are three main sources: the raw material, like in case of seafood, the fisheries, or the fresh products in case of vegetables. Number two, the food handlers, which is present in every food industry. They are a constant source of such type of contamination. And thirdly, the food contact surfaces, where the biofilm is actually they already being present over there. So all these collectively could become the source of contamination in the food industry. Again, I have taken this picture from one of the paper, which is at the bottom of the milk tank, and it again shows the formation of different microorganisms, and they are protected by biofilms. So dairy industry has got raw milk tanks, pipelines, butter centrifuges, pasteurizers, cheese tanks, conveyor belts, trolley wheels. So you can imagine that the bacteria or microorganisms can be present in every nook and corner of the industry. They could be present, they could survive at a very low temperature like psychotrophic, or at a very high temperature like thermophilic. Fish industries, they may, the fresh fish products may suffer from biofilm formation by pathogenic species, Aeromonas hydrophila, monocytogenes, Salmonella, Vibrio. And the poultry industry, which could be the dust surfaces, poultry feed, feces, transportation of live poultry between production and processing units, they could be the constant source of contamination. In the meat industry, again, I have taken this picture from one of the research paper, and I want to show you that the blue field, if you see at your right side, it indicates the detection of bacteria, and the blue fields also show the detection of EPS components. So these are present in the samples which are obtained during operations and after cleaning and disinfection. Even after disinfection, they do present over there. So you can imagine from filling, cutting, weighing, smoking, slicing, packaging, curing, delivering, cooking. So whatever the surface is in contact in the meat industry as well, cutters or conveyors or screw conveyors, they could be the result of the bacterial infection. Ready to eat industry, ready-to-eat foods can be considered as a relatively high-risk food because the products will be consumed directly without undergoing any other process. Refrigerated RTE products, smoked fish, sliced materials, unpackaged or repackaged RTE foods, they are also considered as high-risk foods. they are of major concern. And in this case, E. coli and Listeria monocytogenes, these strains should be of high concern in wide variety of RTE industry. This is the overall table I just made, and I want to show you that several pathogens, their survivability, and you can see the health issues related to this one, and they could be present in any surfaces like stainless steel tanks, pipes, Teflon, glass, PVC, and so on. Now the third, which is the main part of today's talk, is about the prevention of biofilm. So if there are different methods of preventing those biofilms, these could be the chemical treatments, enzymatic disruptions, nanotechnology agents, quorum sensing inhibitors, bacteriophages, biosurfactants, bacterial sins, essential oils, and some of the physical treatments. So the first one is the chemical treatments. Several concentration dependent and time dependent chemical sanitizers can be used as a biofilm treatment. And chlorine is the most widely used in the food industry, like sodium hypochlorite. But resistance to chlorine treatments has compounds like metacots. They are widely used as sanitizers in the food industry, and some of the salicylate based esters are also being used in the industries as the chemical treatments. Second one is the enzymatic disruption. Enzymes are considered as green countermeasure. We call it as green because they are biodegradable and they are low toxic. So these two features make the enzyme technology as a powerful tool for biofilm control. The enzymatic disruption technology is widely used in the food industry because the enzymes you can see in the figure as well, that it degrade the EPS layer, but and then it allows the bacteria to be open in the environment, and then any chemicals can wipe it off. Nanotechnology. Because of the nano size of these materials, it exhibit exceptional features to develop such type of innovative products. In nanotechnology, the main principle is because of a very small size, it has a large surface area to volume ratio. And thereby, because it is exposed a large surface area, it creates a high number of functional sites, and thereby it enhances the influence of nanoparticles on a given microorganism. Previously, silver compounds, they were used as an antimicrobial effects and has been used as well nowadays by using the nanotubes. But some of the metal oxides like iron oxide, titanium oxide, zinc oxide, copper and magnesium oxide are also being used. So what actually the nanotubes do, they interrupt the quorum sensing. They do not allow the microorganisms to communicate with the others. Or they have the antimicrobial nanoparticles in it. And once the microorganisms attach such type of surfaces, they are being dead. So nanotechnology is a good way of controlling these type of biofilms. Then the quorum sensing interruption. Bacteria, because ask other bacteria to come by knowing or recognizing their molecular signals. So if we interrupt the quorum sensing, then the other bacteria will not come and it will not enhance their colony. So quorum sensing regulators can be knocked down by using certain antimicrobials. We have the interrupting metals like nickel, cadmium, and they inhibit DNA and RNA polymerases and thereby inhibiting the quorum sensing. The next is the bacteriophages. Bacteriophages, which is also you can say simply phages, or they are the naturally occurring viruses. They can infect the bacteria. It is an again different type of bacteria. the attractive alternative approach to antibiotics. What they do, they can either coexist with their host by inserting themselves into the bacterial genome, or they destroy the lytic bacteriophage, which is known as lytic bacteriophage. You can see in the figure that viruses, they attach to the surfaces, and then like by adsorption, then they come inside, they inject their nucleic acid, DNA replication, phage assembly comes over there, they replicate, they are so much, there are so many viruses are there, and then those phages are released and causes the cell to be lysed. The next method for controlling the biofilm formation is the biosurfactants. Biosurfactants, these are the natural compounds, and normally they are of microbial origin. Obviously, the word bio means some of the living origin, and they have the ability to modify the hydrophobic characteristics of the bacterial surface. Once they interrupt or modify the hydrophobic characteristic, then the bacteria cannot adhere to any surface, and thereby they cannot make the biofilms as such. So biosurfactants, these molecules insert themselves into the microbial cell membranes and alters their adhesive properties. Bacteriosins. Bacteriosins are the proteinaceous or peptidic toxins, which are again produced by bacteria and bacterial strains. You can see in the figure that at the left hand side, you can see in the absence of bacteriosin, EPS layer is still over there, which already is being made on the solid surface. But in the absence of bacteriosins coated when they are on the surface, EPS layer is being degraded. So nisin is most commonly or widely used bacteriosin, and it is FDA approved, and it is being isolated from Lactococcus lactis, which was able to prevent adhesion and biofilm formation by Listeria monocytogenes. Other bacteriocins which is being used, but that should be GRAS, like generally recognized as safe, particularly if they are used in the food industry. Essential oils, it is again very interesting that some of the compounds which are derived from plants, they have anti-biofilm properties. So these are the essential oils like camphor or eucalypto, limonene. They can be used to control the biofilm, but still it is not very much effective method. High hydrostatic pressure. High hydrostatic pressure is a physical treatment actually, and it is a non-thermal treatment. We don't use heat in it, and it is a disinfecting treatment, and it is used to inactivate the bacterial cells. In this method, the surfaces or the foods, they are subjected to water pressure from 300 to 900 megapascals inside a pressure vessel. And such a high pressure can actually is used to control or destroy the bacterial cells. But the issue with this type of method, that this is not very much effective in the case of endospores. The next one is the non-thermal plasma method. It is again a novel and emerging antimicrobial tool. And in this case, what happens, the gas is being used, which is partially ionized with oxygen, nitrogen, helium, and they are all, and even the UV light, they are all are mixed together at atmospheric pressure, and they work by acting on the unsaturated fatty acids of the lipid layer of the microbial cell membrane, and thereby preventing the transport of biomolecules across it. So decontamination process becomes very easy by this non-thermal plasma method, and it is less expensive. Lastly, very interestingly, probiotics also show the biofilm, anti-biofilm properties. Probiotics are actually the live microorganisms which, when administered in the body, it should be, the strength should be 10 raised to the power 6 to 10 raised to the power 7 or 8 CFU per ml. They have various properties. They are good microorganisms, good bugs, and they also target microbial biofilms. They produce antagonistic substances like biosurfactants, bacterial sins, which we already like discussed before. And all those substances, they hinder the activity of pathogenic bacteria. They also prevent the quorum sensing that does not allow the bacteria to talk to each other. And they generate unfavorable environment to the conditions to the pathogens like pH alteration, surface and nutrient competition occurs, because once the probiotics are on the surface, they will not allow the pathogenic bacteria to come and attach to that surface. So if I conclude my talk, so the overall summary points are: the biofilms are microbial cells that have aggregated together on a surface, and they are held by extra polymeric substances, EPS. Once the biofilm in a mixed culture, could be mixed or could be the single culture, but if they are present in the mixed culture, they indicate that interspecies interaction would contribute to increasing the co-cultured biofilm formation, and it is more resistant. As immobilized cells, they are important and challenging approach to food industry. There are different control measures, so food industry should not focus on the chemical treatments or disinfectants only, because bacteria hide themselves inside the EPS layer, and they can be a constant source of contaminations, and the diseases are over there. Thank you.
Thank you, Dr. Asma Suhail, for your wonderful presentation. Now the house is open for question one by one. We can take 5 questions, please.
Sir, I have a question, if you allow me to.
Ji, please.
Thank you, Dr. Asma, for such a nice talk, and I think you explained very well all the things. My question is related to basically a treatment that is in that one we use mostly. I'm talking about the fruits and vegetables. So we use ultrasonic waves to clean the surface of the fruits and vegetables. So if you can elaborate, or maybe if you can give me some brief idea, is it that effective against this kind of the formation or disrupting the biofilm? If you can please put light on this one. Thank you.
Yes, Dr. Usman, ultrasonic waves also, they are effectively remove the biofilms, particularly in case of fruits and vegetables. Yes, they do. Like I covered only the major one, which is like which we can use, but ultrasonic waves do remove biofilms as well. They have the efficacy of this one.
And the next, please.
Thank you.
Can I ask one question, sir?
Ji, please.
Dr. Aisha Riaz from Faculty of Veterinary Animal Sciences. Dr. Asma, very nice talk. I just want to ask that you gave a few the methods which we can prevent these biofilms. What will be the best in Pakistan scenario? And do you have any idea that in Pakistan, how people are, which method is people are using more? Secondly, in the poultry farm environment, and like you talked about the dairy, what would be the best? I mean, I just wanted to ask that which one is the best for in the Pakistan scenario. Thank you.
Dr. Aisha, actually, normally we use the biocleaning agents or sanitizers to clean our food industry equipments or like the dairy industry, which I told you. But the thing is, as I mentioned in my whole talk, the bugs are actually hide themselves in the layer. So we need to remove that layer. So not only the disinfectants can do, but the combination of disinfectants with any techniques. So it should be done. I'm not sure that all the industries like they are doing this or not. That's why I'm saying that it is a threat to the food industry. Not even in Pakistan, but even globally, the people are talking about these biofilms now. And not only even the food industry or the dairy, you know, the medicinal implants like the plaques or like the dental things they put on, they have got a lot of biofilms over there as well. So in Pakistan scenario, normally sanitizers or disinfectants are used, but the other treatments should be used. They should now focus on the two or the combined treatments to remove those biofilms. And what was your second question? I'm sorry.
Yeah, it's the same. Thank you. It covered the answer both. Thank you.
Any more question, please?
अस्सलाम वालेकुम मैम अस्मा, आपने बहुत अच्छा एक लेक्चर डिलीवर किया है। मेरा क्वेश्चन ये था कि जो आपने बायोफिल्म्स का बताया है रेडी टू ईट फूड्स में, तो वो तो फूड ऑलरेडी प्रोसेस्ड होते हैं और वो पैक्ड होते हैं काफ़ी सारी टेक्निक्स लगा के, तो उनमें फिर बायोफिल्म्स कैसे प्रोड्यूस हो जाती हैं, ये उसमें प्रेजेंट होती हैं? शुक्रिया।
उमर, मैंने ये बताया था कि जो रेडी टू ईट फूड्स हैं वो सबसे ज्यादा एक्सपोज्ड होते हैं क्योंकि वो ओपन एनवायरनमेंट में पड़े होते हैं, उनकी ऑलरेडी शेल्फ लाइफ बहुत कम होती है। तो उसमें जब अगर बैक्टीरिया प्रोड्यूस हो जाए तो उसको तो बहुत ही मतलब हमारा चैलेंजिंग है उसको कंट्रोल करना। तो उसके लिए हमें डिफरेंट जैसे टेक्निक, जैसे ये मैंने आपको लास्ट में जो फिजिकल ट्रीटमेंट बताया था, हाइड्रोस्टैटिक प्रेशर लोग ये बाहर यूज कर रहे हैं, या नॉन थर्मल प्लाज्मा लोग यूज कर रहे हैं, इकोनॉमिकल भी है जो मैंने इसके ऊपर जितनी स्टडीज की है। तो हमें इन चीजों के ऊपर अब फोकस करना चाहिए कि हमें अपने रेडी टू ईट फूड के, और पैकेज्ड हो या रीपैकेज्ड हो, तो हमें सॉरी अनपैकेज्ड हो या रीपैकेज्ड हो, क्योंकि पैकेज दे आर स्टिल सेफ, लेकिन अगर आप उसको रीपैकेज करें या वो ओपन हो तो उसमें आपके पास ये वाले इश्यूज बहुत ज्यादा आ जाते हैं।
Last question, if we have any. I think our questions are covered. No more questions from the audience. Thank you very much, Dr. Asma Sohail, for your wonderful presentation on the biofilm, and thank you for participants for sparing your time for this learning of these students. And I just want to tell that next our seminar is about the artificial intelligence, and the speaker will be from the University Institute of Information Technology. I will circulate, and I hope the most of the students, research students and faculty member will participate. Thank you very much with us. Have a nice time and wonderful holidays.
Thank you very much. Thank you.
Okay, sir.
Thank you, sir.